Hilary is an associate professor and program advisor for the vertebrate paleontology program in the Department of Earth Sciences at Carleton University in Ottawa. She began her training at UTM, followed by a PhD here in Calgary with Doctors Jason Anderson and Tony Russell, working on amphibian evolution. Following this, she completed a postdoctoral fellowship at the Museum of Comparative Zoology before moving to Ottawa, where she has continued work combining fossil data with experiments using modern amphibians to understand their evolution. Hilary's lab conducts annual field expeditions to Atlantic Canada.
In any case, I'll just start with a really brief acknowledgement, just some land acknowledgements here about both where I work, where I do my field work, which is out in the East Coast, as well as where we are today, talking at this conference. And like I mentioned before, most of this work is going to be sort of spurred from what I began doing here in Calgary with Jason and Tony Russell, as well as the supervisors. And some of the students and PDF contributors are shown here. So as you're going to see, various acknowledgements to publications. These are the folks that have been vastly responsible for putting these data into the literature. So where this all began, believe it or not, but the origin of amphibians has been a very hotly debated topic amongst paleontologists. You probably take it for granted when you look at a salamander or a frog today, or a caecilian, like the other group shown up in the top, that they aren't necessarily agreed upon as being each other's closest relatives or not. And then one layer on top of that is there's been a lot of argument and debate about whether or not which group of fossils these animals actually come from. And so when you kind of think about that from looking at the living ones, you're looking at they're all cold, wet, slimy, squishy, small little things. Of course, they're each other's relatives. But when you actually look at them from a paleontological perspective, looking at their skeletons, you can see here that actually this is a bit more challenging than one might possibly think. So of course, frogs, and you can see that little skeleton. Let me see if my laser at least works. Frogs here, of course, are highly adapted to being jumping. Salamanders are pretty generic in their body form, but we give them a lot of credit in other areas. They're vastly diverse in their habits, development, biology, reproduction, all sorts of other ways. And then, of course, caecilians, this third group that which you would probably be the least familiar with, these elongate, limbless, burrowing forms. And inside here, you can actually see these are all fetuses of unborn babies that give birth. Most of them give birth to live young. Very unusual, strange group of amphibians. So when we basically then sort of put those paleontologist glasses on, it becomes maybe not a big surprise that there has been a lot of debate about where these animals come from in the family tree of vertebrates and tetrapods in general. So when I was a PhD student, or just starting out as a PhD student, there were three major hypotheses in the literature, and each were vastly different in terms of how amphibians are related to each other and from which group of fossil amphibians they were coming from. I thought this was very odd, and one thing that you can kind of see is that caecilians seem to be a group that sort of jumped around quite a bit, and they were pretty poorly known. I had only just learned about them myself as being a group of animals that were alive at all. So there was a lot of mystery enshrouding caecilians, and I thought that maybe we could attack this project of lissamphibian origins, or where they're coming from, by taking a closer look at this group of caecilians. And so one way that we wanted to sort of investigate this was by looking for new data, and CT was actually emerging as this really fascinating way of getting new internal anatomy. These are very rare museum specimens. You can't cut them up or dissolve them and do things, but if you actually just pop them in a CT scanner, we can extract tons of new anatomical information that we previously hadn't had access to before. And so one area that was emerging as being really informative is the Braincase. So where the bones that are forming around the brain inside of the skull, that this part of the brain should be pretty conserved and give us a lot of really important information about the evolution of this group. And in fact, that is what happened. We were pretty confident in extracting these data, and we got new phylogenies, and I'll show you what that looks like in a moment. One of the other issues is the fossil record of caecilians has been very sparse as well, and at the time we had basically one representative of a stem caecilian or an ancestral caecilian. And it had been debated between paleontologists and neontologists, the people that study living forms, whether or not this was a true caecilian or not. So we thought, hey, let's CT scan it, take a look at its Braincase, and to see if it looks like a modern caecilian. Sure enough, it did. Looks an awful lot like a modern caecilian Braincase, and in one particular attribute, it's very powerfully caecilian. And that is this little bone here, where basically imagine you're the brain of a caecilian right now, and you're looking out through the snout of the skull. This is basically where the olfactory nerves would punch through the front of the snout into the nasal cavity. This is a living caecilian, and this is the fossil caecilian, the caecilian here. Living caecilians have two pairs of holes here. No other vertebrate that I know of has this pattern, and this has to do with the fact that they have a very unique sensory organ called the tentacle that no other vertebrate has either. And so the way the olfactory nerve comes through the front of their skull, it splits off into two trunks. One goes into the tentacle and one goes into the nasal cavity. Eocene had that pattern. Really hard to see. It's a crushed up fossil, but it's there. So we put all these data together and create this giant, massive, really boring family tree. But this was the first morphology-based phylogenetic analysis of caecilians that actually demonstrated we could improve our understanding of caecilian evolution. And actually, when we included Eocaecilia in this group, we basically then resolved between those three hypotheses as to where amphibians may have come from. Today, the problem continues to exist. However, it's changed a little bit in its dimensions, but I'll save that for another day. So we thought maybe we could look at braincases of other problematic vertebrates and try to see if we can help resolve some of the controversy amongst them. This is a little specimen that is known as a microsaur, one of the animals that was closely allied with caecilians in one of those three hypotheses. So basically, we CT scanned it, took a look at its braincase, and found, yes, it has a lot of similarities with caecilians, but many of those similarities are actually most likely due to the fact that they're both burrowing types of vertebrates. So some of the similarities, including the lots of fusions and co-ossifications of the back of the braincase, a very robust anterior portion of the braincase. However, when you look at the distinct features, general morphology and proportions were quite different, and then as well as the patterns of foramination, where the cranial nerves and vessels are going, were actually quite distinct. So a lot of the similarities linking caecilians with microsaurs, we concluded at the time, were probably just due to functional reasons. Could we actually then resolve problems with another really enigmatic group? This is a group known as the albanerpetids, formerly albanerpetids. This is a group of amphibians that made it very close to the present day. They made it through the PT extinction, but are now extinct. We do not have representatives today. And they have been proposed as either being salamanders, stem frog salamander relatives, stem amphibians in general, or just lissamphibia and citrus. We don't know what they are. So with Jim Gardner at the Tyrrell Museum, we actually had a little brain piece of this albanerpetid and CT scanned it, and I was thinking, oh, I could apply all this new knowledge that I have from looking at extant caecilians and amphibian brains and ears and conclude what albanerpetids are. Turns out, no, it showed a mishmash of features of all of these different possibilities. And so I was like, oh no, but it turns out this group is just deeply, deeply weird because this fossil specimen was found in amber pretty recently. This paper came out in 2020. It is a perfectly preserved skull of an albanerpetid, and even they could not figure out what the heck it was. So it came out as all over the phylogeny again as well. So I'm like, okay, fine, it's not my problem. So there just are some groups that are going to be challenging nonetheless. Switching gears a little bit now. So what I do presently is mostly work on the time period of when all of these animals are coming into their own, basically the origin of terrestrial communities of vertebrates. So this takes us to the Carboniferous, and we've seen some introductions to this time period as well. The early Carboniferous, this is a time when the terrestrial domain is dominated by invertebrates. It's going to take us not too long, but in getting into the later part of the Carboniferous, suddenly we have basically everybody that is present today alive then. So we have at this point in time our earliest records of the reptile line, the earliest records of the mammal line, as well as very early records of the amphibian line. So in a very short period of time, the terrestrial habitat becomes very well populated with vertebrates. And we're trying to understand how did that happen and what are those, what is the level of similarity or ingrained patterns of those communities back then that have been maintained to the present day. So to do that, we go out to the best place on the planet to look at this time period, Nova Scotia. And actually, New Brunswick neighboring right now as well is emerging as another great place to work. So this is where we spend most of our field seasons out in Joggins along the Bay of Fundy. And there it is a very special place because of those trees that we learned about already today, those large hollowed out trees that were either great places as refugia for animals to live. You think today, what's living in a log in the forest? Well, like everything that could possibly live in there. There's salamanders, there's bugs, there's everything. So why weren't the animals doing that back then? Turns out they might have been doing that. So we find our earliest records of reptiles and amniotes in general. So these are the ancestors of mammals, reptiles, and birds, Hylonomus here, found inside of these trees. And that's one of the most important reasons why we probably have fossils out there is because of this unique mode of preservation. We do also, though, fortunately, but very, very rarely get examples of perfectly articulated skeletons on the beach. So these are slabs that were found. Basically, these are two Joggins specimens and Turpentine that you can see at the Joggins fossil cliffs right now. Calnephalon is in my lab right now, but we just finished up with it, so we'll be going back as well. So when you're out there, you can actually say hello to these folks. These are probably the only two articulated skeletons I know of in the history of collecting joggins that have been found. Not good hope the prospects for going out there, but we do go out there every year and we find lots of other different kinds of fossils in different ways. So, like you've seen earlier, this is basically what our targets are when we're out on the beach. We're on the Joggins Cliffs here, basically on the beach. We've got 4-hour windows, as we've learned. We've got basically between high and low tides to get out there, get to your spot, extract as much rock as you can very quickly. If you're lucky, these tree trunks have fallen out on the beach already for you, and you just kind of roll them off. It's not as simple as that, but just roll them gently off the beach in a quad or something, luckily, or a dolly. But this one in particular had a lot of like coaly material. There's a lot of plant material sometimes in these stumps, which indicate maybe there could be some bony material inside there. So we were very interested in this tree in particular. So we hacked away at this really quickly. I'm not freaking out. This is me here. But I am supporting my head, watching, making sure that the cliff above is not going to come down onto my trusty students. So, and Brian here is my collaborator. He's a Joggins resident who has basically found a ton of really important material out there. So yeah, so we're excavating this tree, getting the material out. But yeah, so taking a closer look at some of these historical specimens here. So we're actively looking for new material, but we're also looking back at old material that has been in museum collections for a long time and applying these new techniques and methods to basically see if we can learn a little bit more about them. So this is Dendrocraton, this beautiful skeleton that was found on the beach. And you can see he's entombed in this massively dense piece of iron-rich siltstone. This thing, this block weighs about 40 pounds. It's only about this big, and I took that thing to Texas if it was going to kill me. 50-pound case on my shoulder. I tried to make it convincingly look like carry-on so they wouldn't make me check it. And didn't make it through, almost made it through the X-ray CT scanner, get it on the plane. Robertson had screwdrivers in, and they couldn't open the box because it was a Canadian invention, I don't know. And so now I travel with a Robertson head bit in my pocket. And anyways, long story short, we got this thing CT scanned, and for the first time we got to see basically the whole face of this little beast. Here's what the surface looks like. So we had one whole side of its face had been trapped in stone, but finally, for the first time, we could see its full little smile. And it's quite a beautiful little specimen. And Dendrocraton had been sort of bounced around. It was being sort of its position within the Temnospondyli tree had moved around a little bit. And so this is really important for gaining a bit more insight into where in that Temnospondyli tree it actually resided. And so with the power of CT and being able to basically digitally dissect the skull into multiple pieces, we revealed some really fascinating new anatomy, including these massive fangs on the palate that were hidden and holes in the jaw where these teeth were slotting into. And these are very ancient sort of primitive traits. And this basically resulted in an analysis where the specimen fell a little bit deeper in the tree of Temnospondyli amphibians. Calignethalon, another really cool animal. These are an enigmatic group known as embolomeres, sort of amphibious-like forms. They've been jumping around in the family tree of life as well quite a bit. This specimen was found by some children in the 90s. They're now grown-ups. So I keep calling them found by kids, but they're obviously now totally grown-up people. But so like I mentioned, embolomeres have been a little bit problematic recently. And here's a paper that also came out of Calgary, out of Jason's lab back there as well, by another Jason Pardo. And he was also looking at Braincase anatomy in this group down here, that basically resulted in moving a whole plate of animals from way up in the tree to way down in the bottom of the tree. And that had to do with sort of, again, these Braincase features. So what was happening, my Protogeronus didn't show up. So the embolomeres had previously been coming out very high up in the phylogeny, close to the amniote origin. However, even in this analysis, by looking at Braincase traits, embolomeres were falling down quite a bit more deep into the phylogeny tree of life. So now we have this perfectly, beautifully preserved specimen of an embolomere. If we CT scan it, can we extract more information now again to help resolve some of the mysteries of surrounding where embolomeres should fit? Should they be on the amniote stem or on basically down at the base of tetrapods, period? This is a pretty huge consequences for the, understanding the evolution of traits. So we got really nice Braincase anatomy, beautiful forelimbs that were otherwise buried in the rock, as well as some new dental features, palatal features. And, this basically all kind of comes together to show us quite a bit of modifications within the amphibian part of the tree of life. So on the one hand, we have, the issue of caecilians as being, mysterious as to whether they were up here closely related to amniotes or not. The CT work is sort of anchoring them back down here a little bit deeper within the temnospondyls. Eocecilia as being a little bit more firmly rooted on the caecilian stem, and there's a lot of implications about that, which we won't talk about today. Microsaurs being, having their own position within phylogeny, and that any similarities with caecilians are likely due to their convergent functional habits. Albanerpetids, we don't really know where they belong. I think they probably should maybe be on the stem of lissamphibians, but that can be a discussion for another day. Dendripodon here, we have him sort of rooted within the Temnospondyl tree, but maybe actually should be falling out quite a bit more deep within the Temnospondyl tree. And then, of course, what was going on with that embalmir, Calnethlon, well, maybe they belong way down here instead, maybe a little bit higher up. Our analyses are putting them a little bit higher up, but there's still a lot of work to be done there. So what does some of the new material look like now? So we're out there every year. It's been a couple of years. There's been a pandemic. There was a human that came out of me. I didn't go in the field. I was not a good pregnant person. And walking on these cobbly beaches was going to be too challenging. In any case, this is some material that we've gotten out of Cape Breton recently. So one of these trees produced a whole bunch of animals. One of these that we've already worked on is this beast up here. This is a Baranopid synapsid, so more closely related to us. And it was found in the tree trunk at the base of the tree. It's really hard to see here, but there's a little baby skeleton, and the tail of this animal wraps around. And it was found sort of looking like it was in a denning posture, such that we propose that maybe this is an example of parental care or extended postnatal care. The oldest example in the tetrapod fossil record. Might have been crazy to think this was the case. However, it is a member of the same group that was the current record holder. So there is a specimen from South Africa that is very obviously a large individual with 6 juveniles under its limbs in the same family. So we think, okay, this probably might have been an early experimentation with postnatal care. But I will show you very quickly what's going on with this little beast down here. This is one of our new specimens that we're about to submit. Might not look like much in this picture, but this is basically a punctiliid microsaur, known more well from this animal known as punctilus, that is an early Permian form. This animal is quite large, probably one of the largest microsaurs that we have from the Carboniferous now, and it is of this group otherwise pretty much known to be only Permian in origin. We CT scan this. So what we're looking at now is a transection through its snout. And what the arrows are pointing at are like a set of jaws inside of its mouth. So the marginal dentition would be along the edges here. So whether, our dentary teeth and the maxillary teeth along the edge, but inside the mouth of this animal are teeth packed close on the palate and opposed on the lower jaw. We'll take a look at what this looks like in a little bit more detail in the CT data. Here's the skull of this animal CT scan. My student actually undeformed it. She's a lot more talented than I am with CT data, such that here we can now actually have a little image of what this skull looks like in 3D. And you can see it peeking into the jaws inside. It's basically got 2 mouths inside this little head. Flipping around, some more things. And and then here is a closer look at those internal mouth dentitions. So on the palatal surface, this yellow bone up here, and these are the dental teeth on the lower jaw. And I think this video is going to show basically this is the outer marginal teeth. We're going to strip those away. And now we're looking at basically a whole new set of jaws inside of the mouth. And what these basically look like are dentitions for animals that arise in the Permian that we interpret as herbivores. So basically these are occluding batteries of teeth that probably were used to crush and grind the tough plant matter that was basically prevalent at the time, and maybe one of the earliest examples of herbivory. Of course, we can't rule out this is probably chewing other hard things, or if it encountered bug bits, it was probably able to blast through those as well. But there is actually very nice, closely related specimens that have basically tooth wear that show that these were sliding past each other as well, these little tiny teeth, some abutting and some grinding past. Maybe we even have some early examples of herbivory. So I hope I haven't gone too over, but basically just to summarize, applying these CT approaches has basically opened up a whole new treasure trove of data that are basically allowing us to attack old questions, propose new questions, use old data, apply it to new data, and we're basically able to revise some really cool evolutionary transitions in the early evolution of vertebrates on land. So that is all I have for you today. We do have time for some questions.
The caecilians, did they breathe through the skin or did they have lungs? I understand that salamanders did both. Some had lungs, some breathed through their skins. But what about the caecilians?
They have lungs. Yep, yeah, they have lungs, so they're air breathers. I mean, all amphibians, because the thinness of their skin will permit what's called cutaneous respiration. So there will be some exchanges of gases on the skin surface, but they aren't exceptional skin breathers. I mean, you can suffocate them. I mean, you don't do that, but I think you could suffocate one. So they wouldn't, yeah. That's a test.
Thank you.
I'm curious, in the looking for things in the tree trunks, you're picking on what looks like 10 meters worth of tree. Do you find them only near the bottom, or can sediment fill and then they get filled, or they're only near the bottom and so you go for the bottom and spoil the whole effect of the nice tree and disconnect it from the roots?
That's right. Yes, so we spoil the whole tree. No, I'm just so those trees, as you know, I mean, the erosion rate is so massive at Joggins that those things are going to come out within a year or two, and they're going to be taken out by the tide. So what's more important for us is to get at them quickly, get the material out, and save the false material. So, but what we have been finding, so in the past, we would target the bottoms because that's where the animals we think were most likely occupying and living. However, in the tree material that we're getting out of New Brunswick now, there is stratification and there are multiple horizons of bone material throughout.
So you've actually seen bone material at different horizons.
That's right. Yeah. There's like stratigraphy inside the tree trunks.
Right. No.
Yeah.
There's some argument about whether they were hibernating or estivating in them or whatever. And then there's other stories. And from the look of that, I think they were just falling in them and trapped forever.
Yeah, that is definitely one hypothesis. I think basically probably a combination of everything must have been happening. There's definitely instances where there's so much disarticulated material in there that it looks like it was just washed in and trapped in there. The case of the Varanobid with the juvenile, though, those bones are minuscule and tiny gastralia are preserved all in place in articulation. That animal had to have been in there, killed, and buried extremely rapidly. So I think it was probably living in there. It didn't get washed in. So, yeah, and in that case,
do you think was living in there thinking it could get out in spring or whatever?
Well, we're in the tropics at this time, right? So we're in a tropical time period. There's not a lot of seasonality per se. There's wet and dry seasons, but I think they were just doing a whole bunch of everything. Yeah.
So how large exactly was like the new microsaur you found?
Yeah, so it's about the size of Pentilus itself right now. Our big skull. We have some smaller skulls as well that are coming out of New Brunswick, but it looks to be very close to the size of Pentilus itself, some of the large. So like the skull is about, so this is a giant microsaur, okay? Microsaurs are called microsaurs because they're very tiny reptile-like animals. And so this is sort of a bit of a misnomer, but it's a giant, so it's the skull like that. So the animal could be like snout bent, maybe like that, throw a tail on it.
I don't see any more questions. Well, thank you very much.
Thank you so much, Lauren.
Thank you.